and we're really helping clinical stage biotech companies to overcome the limitations of off-the-shelf equipment. I think it is about being quite smart about what to automate when. I'm really looking for companies who are trying to do something a bit different.
Onyx Live is proudly sponsored by Humphreys Law and Panacea.
I'm Stuart Lowe. I run the Advanced Therapies team at TTP. We're a group of scientists and engineers, and we're really helping clinical stage biotech companies to overcome the limitations of off-the-shelf equipment. When there's a point you find that the cost or the throughput or the consistency isn't quite what they desire, you can actually go and develop a custom piece of equipment, and that's what we're helping them to do.
Okay, and so those bottlenecks, why exactly did they arise in the first place?
So there's a few reasons. The main one is around a need to prioritize clinical data in the early stages. That's what the regulators want to see. That proves the science, that brings the investors on board. But it bakes in some commercial risk, i.e. the processes that you use to generate early clinical data might not be the best processes for manufacturing at scale, manufacturing at commercial scale. It might cost too much, for example, it might take too long.
And is that something that we could look to change in the future, or is that just the nature of the game?
There's a few things you can do. You can actually think about modelling the cost of goods quite early on and saying, these are actually the points in my process where it is going to be very sensitive as I increase the number of doses. You can then say, well, I'm going to target this part. Maybe it's the most labour intensive, maybe it's the most variable part of my process, and say, well, we'll make a tool that actually really helps with that part. So you're not, there's different options. There's kind of saying, well, I can go all the way to full-blown automation end-to-end, or I can stay manual, but you can go somewhere in the middle, which basically says, gradually introduce automation as your process evolves. And so I think there is a space for that. We're working with a few companies who are trying to bring new types of product to market, new personalized therapies to market, and they found this really useful because it allows them to move from kind of isolators in a category B environment into standalone pieces of equipment where you can do multiple patients in a class C environment instead.
One of the great beauties of automated manufacturing is reducing the cost of cell and gene therapy, but then I suppose there's a barrier to entry here in that automation equipment itself is very expensive, and so how far do you think we are in the process of making that affordable and accessible to everyone? It's
not just the cost of the equipment either, it's the cost to change the equipment, which is a big factor that actually adds into people getting over that kind of cost hurdle. So you spent a lot of money on some equipment, you've developed a process which kind of serves your needs up to a point, and then in order to take advantage of any improvements, you may have to do another comparability study, you may have to do more validation, and that basically locks you in to a choice that you made really, really early on in the development. So kind of answer your question, where do we go with the cost? I think it is about being quite smart about what to automate when, starting small, targeting areas with the kind of highest risk, which allows you the greatest flexibility as your process develops.
And is there any kind of forward compatibility with this legacy hardware compared to modern automation, or is it really a case of ripping out everything and starting afresh?
We are seeing some examples where you can actually adapt existing equipment. So we spun out a company called Cellular Origins a few years ago, and their thesis is actually to actually take the existing hardware, modify it so that it fits within a robotic framework, and now you can actually say, well, I'll keep my legacy equipment. I need to adapt to the consumables for robotic handling. I need to actually have a way of piping data in and out of the equipment, because a lot of the legacy equipment is actually designed for manual operation. So you punch in the parameters, you read out things, you load them manually. So there is a way to retain legacy equipment, but it does require quite a lot of investment then in robotization. Whereas what we're trying to do at TTP is kind of saying, yes, you can do that, but maybe it might be an earlier opportunity to say, can I do something different that's tailored for what I want it to do, what the biology needs it to do.
Gotcha. And how's advanced therapies going so far today? Who have you been speaking to?
Yeah, so I've had a few meetings. It's kind of fast and furious here at Advanced Therapies. You can see it's a very busy show. You bump into a lot of people you know already. So I've had a few conversations with contacts in kind of AI-driven media optimization. Somebody I was talking to about kind of media manufacture. I've been talking to some of my contacts more in the kind of biotech space about, how are they envisaging the next generation of their processes? They already have commercial processes, but what does the next thing in the pipeline need? I'm really looking for companies who are trying to do something a bit different. Probably not CAR-T, probably more on novel cell types like TILs or NK or macrophages, where some of the processing challenges haven't quite been solved by your standard off-the-shelf manufacturers.
And if you had to hazard a guess, what do you think the next step of that pipeline looks like?
I think what you'll see is a lot more modularity. There are processes which tend to tie up equipment in different stages of the process. Well, I'm going to call out Prodigy because a lot of people know of it, and it's an example where you've had to couple the cell separation with the incubation, and yet the cells spend a lot more time in the incubation than they do in the cell separation. So, next generation equipment, and I'm sure Miltenia are thinking about this as well, is how do you kind of isolate the cell separation part so you can have that much more utilized, equipment occupancy being a lot higher, and then use a lower number of incubators because that basically matches the flow of the process. Gotcha. I think equipment occupancy and footprint of facilities and operator utilization are going to be people are going to be looking at those ingredients quite a lot because they do actually add up a lot to the cost, especially once you kind of get over that point of saying, I'm not doing 100, this isn't an academic process, this is now a commercial process. It doesn't make sense for all of my floor space to be occupied by pieces of equipment that aren't really being utilized 100% of the time. So I think that'll be a shift.
Exciting to look forward to, Stuart. Thank you so much for your time.
Cool, really nice to meet you.